May peace be on all of you. Myself, Dr. Majid Khan, Associate Professor, Loknete, Dr. J.D. Pawar College of Pharmacy, Kalwan, Nasik. I welcome all of you in Anticancer chapter. In this lecture, that is in lecture 3, we will cover antimetabolite and that pyrimidine analog. In previous two lecture, we were cover the alkylating agent, which is a first major class of anticancer. Now the second major class of anticancer is antimetabolite. So in there are three types: pyrimidine analog, purine analog, and folate antagonist. Now in this lecture, we will cover antimetabolite of pyrimidine analog. Learning outcome: after completion of this topic, student able to define antimetabolite and remember the classification of antimetabolite. Understand the activation, mechanism of action, and metabolism of antimetabolite. So what do you mean by antimetabolite? These are the compound closely related in structure to a cellular precursor molecule, and these substances are capable of preventing the proper use or formation of normal cellular product. These antimetabolites are similar enough in structure in many cases to interact with normal cellular process, but differ in manner sufficient to alter the outcome of that pathway. These drugs actually prodrug and require activation in vivo to yield active inhibitor. The administration of purine and pyrimidine antimetabolite require the formation of nucleoside and finally corresponding nucleotide. The antimetabolite and its transformation product may inhibit several different enzymes involved in tissue growth. These drugs are cell cycle specific with activity in S phase. So antimetabolite are the drug which having structural similarity with cellular precursor. So as we know, DNA and RNA made up of bases, sugar and phosphate group. So in bases, that is DNA, RNA bases, there are two types of bases: purine bases and pyrimidine bases. In case of purine bases, adenine, guanine is the example. In case of pyrimidine bases, uracil, cytidine is the example. So here it is a base, that is guanine. This one is sugar attached. So this base plus sugar, it is called as nucleoside. So the base and sugar is called as nucleoside. When phosphate group is attached, then it is called as nucleotide. So in short, nucleoside means base plus sugar, and nucleotide means base plus sugar plus phosphate group. So these antimetabolites having the structural similarity with these bases. For example, here it is a cytidine, which is a normal base of DNA and RNA, and it is a drug, that is Cytarabin arabinoside. So here there is a structural similarity between these two. Similarly, adenine is a base and this mercaptopurine is a drug. So these antimetabolite having the structural similarity with cellular precursor molecule, and these drug like a prodrug require activation. So here activation means in drug sugar molecule is attached and phosphate group is also attached as in a normal basis of DNA and RNA. So these antimetabolite drug require activation like a prodrug, and therefore it insert into the DNA instead of normal bases, and therefore disturb in the DNA and RNA synthesis. So it is the definition of antimetabolite drug. So here in the classification of this type of antimetabolite, there are three classes. First one is pyrimidine analog, and that 5-fluoro uracil, cytarabin, capecitabine. Second, purine analog, 6-mercaptopurine, thioguanine, vidarabine. and last one is folate antagonist, example methotrexate, trimethotrexate. So in this lecture we will see the pyrimidine analog, and that example is 5-fluorouracil. So as we see it here, there is a uracil which is a base for RNA. So this drug having the structural similarity with this uracil, 5-fluorouracil, only the difference is fluorine. So here it is a uracil which is a normal base, and it is a drug, anti-cancer drug 5-fluorouracil, which having the fluoro group on 5th position. So here the structural similarity between uracil and 5-fluorouracil. So 5-fluorouracil, regarding their mechanism of action, 5-fluorouracil is converted into their active form, that is fluorodeoxyuridine monophosphate. Then this FDUMP activated form inhibit the important enzyme, that is conversion of DMP to TMP, thymidine monophosphate, by the enzyme thymidylate synthase. So this enzyme is inhibited by this drug. Therefore, the conversion of DMP to TMP not occur. This step is important in DNA synthesis. So this reaction we already seen in sulfonamide. So it is the summary of mechanism of action of 5-fluorouracil as a pyrimidine analog. So first, 5-fluorouracil converted into their active form called as FDUMP, fluorodeoxyuridine monophosphate. This activated form inhibit the main reaction, that is conversion of DUMP, that is deoxyuridine monophosphate, into thymidine monophosphate by the enzyme thymidylate synthase, and this reaction is inhibited. Therefore the DNA synthesis also inhibited. So normally it is a reaction DMP converted into TMP by the enzyme thymidylate synthase. So if we see this reaction, there is only difference is methyl group. So here there is only methylation taken place. So DMP converted into TMP by this enzyme thymidylate synthase, which contain methyl group on 3rd position. So how this reaction occur normally? So mechanism of this reaction, that is mechanism of conversion of DMP to TMP, is as follows. So it is a mechanism how DMP normally converted into TMP. So here there are 3 components. First one is enzyme, that is thymidylate enzyme. Second one is precursor, that is starting material DMP, uridine monophosphate. And this one is cofactor. This one is cofactor, that is tetrahydrofolate. So here there is a reaction between these 3. So in first step, enzyme, that is thymidylate synthase enzyme having the sulfhydryl group, SH group. Here the S minus act as a nucleophile. This S minus attack to this ring carbon, and this double bond attack to this carbon, and this positive charge on nitrogen is neutralized. So here there is a N double bond CH2. Here is double bond is present. So this bond of aromatic ring attack to this CH2, and charge is neutralized like this. So here enzyme is attached, here attack enzyme. So here enzyme is attached. Then this pi bond of this ring attached to this CH2. So here is there is a bond. So here there is a attachment. So first step, S minus attack to this carbon, this pi bond attack to this CH2, and this pi bond is break and it become charge is neutralized. Then is second step. In second step, this H hydrogen is removed and this bond is shifted here. So here the bond is shifted and this bond is break. So this nitrogen carbon bond is break, it become N minus. So this nitrogen become N minus. So in second step, removal of hydrogen from here, then this bond is shifted here, it become double bond. So it becomes a double bond CH2 like this. So it is a double bond CH2, and the carbon CH2 and nitrogen bond is break. So here there is a this bond is break, and there is a N minus. So here is N minus. Then next step is, that is the important step here, properly see here. So here N minus converted into double bond, here follow the arrow, and this hydrogen or this bond shifted here. So this hydrogen is shifted here, this double bond shifted here into this ring. So we get three product. First product is this one, second product is free enzyme, and third product is, that is TMP, deoxy TMP, thymidine monophosphate. So here DMP converted into TMP, only methylation reaction occur. So here the methyl group is supplied by cofactor. So if we see the reaction, this CH2 is transferred to DMP, and again hydrogen is also transferred to this molecule, so it form TMP. So in short, cofactor, that is tetrahydrofolate, is a supplier of CH3 group. So only methylation reaction taken place. So again mechanism repeat. So here first step is S minus attack to this ring carbon, pi bond attack to this CH2, then this pi bond is break, charge is neutralized. In second step, hydrogen is removed from here, and then it is shifted to here, it become double bond CH2, then nitrogen carbon bond break, it become N minus. Then it is an important step, H from this removed N minus converted into double bond, then this H is transferred here, it becomes CH3, and this pi bond transferred to this ring, so it forms three product, free enzyme, side product, that is called as dihydrofolate. It is a tetrahydrofolate, it is a dihydrofolate, and the final product is DTMP, thymidine monophosphate. So here there is a conversion of DMP into TMP, and cofactor is required, tetrahydrofolate, which acts as a source of CH3 group. So it is the normal reaction how DMP converted into TMP. Then our drug, that is 5-fluorouracil. So it is a 5-fluorouracil. So first it activated into their active form. So as we discussed previously, normal bases contain normal DNA RNA contain bases, then it is converted into nucleoside and then into nucleotide. For example, if adenine is a base, then after sugar attachment it forms the nucleoside, and then after phosphate group addition it converted into nucleotide. So here similar here, 5-fluorouracil after addition of sugar molecule it converted into nucleoside, and then phosphate group is added, it converted into nucleotide. So here 5-fluorouracil, the active form is 5-FDUMP, fluorodeoxyuridine monophosphate. So here is the activation of this drug after addition of sugar and phosphate group. Then next is how this 5-fluorouracil actually act. So we seen the normal mechanism of DMP to TMP. So here instead of DMP, instead of DUMP, 5-fluorouracil taken part in this reaction because it having the structural similarity with DUMP, that is uracil. So here cofactor is same, enzyme is same, but only the difference here is instead of DMP there is a drug 5-fluorouracil. So first step occurs similar manner as in normal reaction. So here again S minus act to this ring carbon, this pi bond attack to this CH2, this charge is neutralized. So first reaction, first step is same as a normal process, but in second step there is a, if you see the second step in normal reaction here, so here there is a removal of hydrogen. So here this step is important. So here hydrogen is removed. From where hydrogen is removed? DMP. From DMP this hydrogen is removed. In second step this hydrogen is important, but in case of 5-fluorouracil there is a no hydrogen. So in case of 5-fluorouracil, instead of hydrogen there is a fluorine. So therefore the second step not possible. So mechanism of action of 5-fluorouracil, here in this reaction instead of DMP drug is there, cofactor is same, thymidine enzyme is same. So first step occur as same manner as normal reaction, but second step is not possible because for second step hydrogen on fifth position is important, but in case of 5-fluorouracil, instead of hydrogen fluorine is there, therefore no further reaction, and therefore the conversion of Dmp to Tmp is inhibited, and therefore there is disturbances in DNA synthesis. So it is the mechanism of action of 5-fluorouracil. Now metabolism of 5-fluorouracil. So it is a 5-fluorouracil. So 5-fluorouracil first. So here there is a oxidation, hydrogen is added here, pi bond is break. So here is addition of H2 by breaking of pi bond like this. So it form the fluorodeoxy dihydrouracil, 5-fluorodihydrouracil, it is this product. Then after that there is hydrolysis, so this pi bond is break, and then OH is added here, hydrogen added here, so it become acid, that is fluorouridine uh, propionic acid. So here there is a hydroxyl, so this bond is break, water molecule is added. Similarly here again in next step, here is hydrolysis, this bond is break, hydrogen is added here, OH added here, so it form, then it form fluoro beta alanine, it is a fluoro beta alanine. So it is a catabolism or inactivation of 5-fluorouracil. So 5-fluorouracil first step oxidation, hydrogen is added, then pi bond is break by the enzyme dihydropyrimidine dehydrogenase, then hydrolysis here by the enzyme dihydropyrimidine aminohydrolase, it form fluorouridine propionic acid. There are 5 carbon, sorry, uh, 3 carbon, 1 2 3 propionic acid, then hydrolysis here, then it form fluoro beta alanine by the enzyme beta alanine synthase. So the metabolic activation of fluorouracil required to produce the anti-cancer effect for no more than 20% of administered amount of drug is most patient. Catabolic inactivation via the normal pathway for uracil consume maximum fluorouracil follow this pathway, that is catabolism. Only 20% require or used by the body as a anti-cancer drug for their mechanism of action. So whatever you administer the dose of 5-fluorouracil, only 20% dose of drug is responsible for anti-cancer effect. 80% drug follow this inactivation pathway. Then second drug of this category is capecitabine. So it is a capecitabine which having the structural similarity again uracil. So it is a capecitabine. So first here there is a activation. So here capecitabine is activated. So how it is activated? So first here there is a hydrolysis taken place. So here this bond is break, hydrogen is added here, it become NH2, and this part is separated as a acid. So here is NH2. Then again here there is a attack of water molecule by the enzyme cytidine deaminase. So here removal of amine group. How amine group is removed? So here first OH attack to this carbon OH. This pi bond is break N minus. Then hydrogen from water attach here, it become NH. So here NH, and this one is OH attach here. Then from here NH2 group and hydrogen from OH group is removed as a NH3. and there is a formation of ketonic group. So it is a formation of 5-deoxy-fluorouracil. Then this converted into 5-fluorouracil by removing of this sugar molecule. Then it converted into 5-fluorouracil. So capecitabine is also anti-cancer drug. It require activation. It is converted into their active form as a 5-fluorouracil, and then this 5-fluorouracil act as an anti-cancer as we seen in previous slide. So capecitabine is a second drug of this category used as an anti-cancer. Therefore, it require also activation. So by this mechanism, this drug activated and converted into 5-fluorouracil. So again, here hydrolysis is taken place. It become NH2, and this acid part is separated as a byproduct. Then here there is a removal of this NH2 group in the form of NH3. So here this on this carbon OH group is attached, then this pi bond break N minus, then hydrogen attached here, that is become NH. So here OH is attached, here is NH, pi bond is break, then removal of NH3. So this NH2 is removed, and hydrogen from hydroxyl group is removed, and this oxygen is converted into ketonic group. And finally, there is a removal of the sugar molecule by the enzyme thymidine phosphorylase to form the 5-fluorouracil. Next drug is cytarabine. So next drug of this category is cytarabine. So here again also there is a structural similarity between cytarabine and cytidine. So it is a cytidine is a normal base of RNA. So it is a cytidine. So it is a drug that is called as cytosine arabinoside, in short Arac. So if we see the structure similarity, so here there is only one difference is this OH. In case of cytidine, this OH is below the plane, but here there is a above the plane. So cytarabine structure is based on the cytosine structure. Modification of normal ribose has produced cytarabine. The cytarabine contain arabinose sugar instead of ribose, and only difference in structure is the epimeric hydroxyl group at position 2 of pentose sugar. The epimeric sugar is similar enough to the natural ribose to allow arabinoside C to be incorporated into DNA, and its mechanism of action may include slowing of the DNA chain elongation. So here cytidine, it contains ribose sugar, which is a normal base of RNA and DNA. So it contains ribose sugar, but here the drug that is called as cytarabine drug or Arac. So this Ara is designated for the arabinose sugar. So it is a arabinose sugar. This one is a ribose sugar, but here is a arabinose sugar. So the difference between ribose and arabinose sugar is only the epimeric hydroxyl group. In case of arabinose sugar, hydroxyl group is on second position. It is above the plane, but here it is a below the plane. So only the difference of this hydroxyl group on second position above and below the plane. So cytidine contain ribose, but this drug, anti-cancer drug cytosine, contain arabinose. Therefore, it is called as Arac. So it having the structural similarity with cytidine. So this drug involved in the formation of DNA or RNA instead of cytidine. Therefore, there is an inhibition of DNA and RNA synthesis. So all these drug, it is a pyrimidine analog. So these drug having the structural similarity with bases. So in this category, we will see three drug. We will see three drug. First one is 5-fluorouracil, second one is capecitabine, and third one is cytarabine. So mechanism of action of cytarabine. So again here there is a activation. So it is a cytarabine drug. So here phosphate group is added. So it converted into nucleotide. So it is the drug without phosphate group, but after activation, for activation phosphate group is added, that is it is a nucleotide, and then this nucleotide incorporated into DNA instead of normal bases. So it form the faulty DNA and inhibition of DNA strand, and therefore it produces the anti-cancer effect. So it is about the pyrimidine analog as a anti-cancer. In next video, that is lecture 4, we will see antimetabolite other classes, purine analog and folate antagonist. If you like the video, then subscribe the channel. Thank you very much.